Okay, you can see my screen, right?
Yes, sir.
Yes, okay. So in the last class, we were discussing about vectors, and we discussed a few things about plasmid vectors, and we can repeat them again, but we will discuss that at a later time. I mean, blue-white screening is done, so we will discuss about PBR322 just in case you have any issues or you couldn't understand from the previous videos. And we also discussed about some basics of phage biology, lambda phage biology, and then we have seen how lambda phage DNA can be used as vectors. And then we discussed a few concepts which are from biology and how they are applicable in cloning larger fragments using lambda phage vectors. We saw the significance of cause site. We also discussed about the space constraints within the DNA that can be packaged into a head particle. And that is where we discussed that how insertional vectors can only be used for cloning smaller fragments, somewhere around 5 kb fragments. 5 kb fragments can also be done in plasmids. Then why use it for use phage vectors? Because plasmids are actually much more simpler to handle. So then the other one we discussed is about the replacement vectors, and they have a very good advantage. To also we discussed about why should we even go for phage vectors. It is to increase the size of the fragment that can be cloned. And then we discussed about the replacement vectors. We discussed what is the importance of left arm, right arm, and so on, and also the stuffer that should be replaced. And we discussed about SPY phenotype. We will review that again today. And we have discussed about the gene library construction using EMBL3a. In this, I will repeat mostly this one today. So hopefully, if you understood this one, it is easy to understand everything else. So the point of going there is that it is something like this. If you have trouble walking here, the concept is to walk higher so that you will feel comfortable in this one. That is how we should see it this way. So EMBL3a is a fantastic subject to study because it includes many. It is convergence of multiple phenomena, and the first one is, or if we want to look at it, it's always important that we see it in this slide as it described yesterday. It is about how the design of the vector itself. Why is it made like this? Why did they have these polylinkers here? And why is it in this order? SAL1, BAMH1, EcoR1. And on the other polylinker, on the other side of the stuffer region, this is the stuffer region. It is EcoR1, BAMH1, and SAL1. And there are also two more things that we need to discuss is why do we have red and GAM, right? So they designed it in this way to reduce the non-recombinants. Non-recombinants, meaning that we don't want vector-vector religation. That is why they designed the polylinkers in this way. They have put in red and GAM to capitalize on the part of SPY phenotype. SPY sensitive to P2 inhibition. And these are the how to use these polylinkers and SPY phenotype, and the whole protocol or the sequence of steps is what we are going to review today and now. As I also described yesterday about BAMH1 and SAU3A, they are isocordamers. That means they will leave identical overhangs. Their recognition sequences are not identical, but they would leave identical overhangs. BAMH1 is a 6-base cutter, whereas SAU3A is a 4-base cutter. We will also add on from what we discussed in the last class. We treat the genomic DNA with phosphatase. And so basically, I'm trying to review this part as two topics. One is for phage vectors, and second is for gene library or genomic library. Let's go. So the idea or the goal is to construct a genomic library. Let's just go to the previous. So I'm going to explain what is meant by genomic library, and for those who did not have enough introduction from previous classes. So you can say genomic library or gene library. We will get to this topic somewhere else again, so we will discuss this again at that point. But it's a good idea to go with the flow at what we are discussing at this point. The first thing is, say, for example, there is a genome. And the genome has several genes in a particular order. It has A, B, C, D, E, and F. And if we can make if we say a genomic library, what we would mean is that we should have constructed several clones, somewhere like this, and cloned in the fragments there. One should have had A, one should be having B, C, D, E, and F. That is a gene library or genomic library. What you mean is, so if you want to define from the concept that I have illustrated as a figure here, then we should say it is a collection of clones. It's a collection of clones representing the whole, representing all the genes. So here I'm being partial and just talking about genes, but all genetic elements, which would include genes, regulatory elements, intergenic spaces, and so on. Elements at least once. That would have been the ideal genomic library, right? If we had only, say, for example, if we did not have this D, then is it a gene library? It is gene library for that particular organism, but it's not a complete one. It is missing some parts. So I hope you understood the definition of it. A gene library is a set of or a collection of clones that would be representing all the genetic elements at least once. Ideally, it is usually more than once. Okay. Why are we saying once is, if we take a genomic library, a genome, you'll take multiple copies of this genome. When you collect from several thousands of cells, and you get the genomic DNA. Right? So A is represented multiple times, and each of these genes are represented multiple times in all the genome as a collection of the DNA or the genomic isolation that you have done. And then you fragment it, and then you try to clone it in plasmid or any of the vectors. So when we practically do what I have described here is a theoretical one. That is the ideal situation. When we do practically, some of them may get cloned often, some of them may not be cloned often. Okay, that is meant by a genomic library, where you should ideally have all the, it's a library of clones consisting of genes representing all the genes at least once. Okay, I should put this. So now, sorry, we'll get to this again. Don't worry. We have to discuss about genomic libraries, cDNA libraries that will come later. What we are trying to do here is trying to learn the same thing, how to clone it, and we are trying to use it for two purposes. We already had good enough introduction to EMBL3a. Sorry, one second. Okay, so we are trying to construct genomic library using EMBL3a. That is what we are trying to do today. So the goal is, of course, twofold. One is to review EMBL3a, and the other one is to introduce genomic libraries. Okay, we'll go step by step. So the first one is, yesterday I did not focus on the genome side, this part. Today I'm going to focus on that as well. So the first thing is, the objective here is, we have a DNA genome. We are supposed to make gene library. Assume that it may have gene A, B, C, D, all, and several thousands of genes actually. When we do that, we are supposed to say, for example, the genome is very large. Should I use a 6-base cutter or 4-base cutter or 8-base cutter? What do you think will be the ideal one? Anybody? If we use 8-base cutter, the probability with which it cuts, probability will be lesser, right? Larger sequence recognition. I think we have done this when we were discussing about restriction endonucleases. If you have 4 bases, what is the frequency with which, theoretically, what is the frequency? It comes to about 256.
256.
That's right, 256 bases. Every 256 bases, it is likely, okay? If it is 6 bases, then it is about 4096. Is
4096.
okay, that's right.
And then 8 goes on. So 4 base.
For 8, tell me the number, please. So I will write, we can all understand them. Anybody? Calculate if you need. Is somebody doing?
Somewhere around 16 million something.
Okay, no, it won't go that big, I think. But anyway, just check it out. That is huge. Okay. So basically, you have a very long DNA and you want to cut it. And if you want to cut it, how big should it be? How much? What should you do? If you choose something that is very rare, then you're not cutting the DNA enough, right? You can choose 6-base cutters, where you can probably then you will have approximately this, but there is no guarantee, because as we have seen one of the figures, we know that the base composition or base distribution composition is also different. GC content is different among different organisms, and also you have different. The distribution within the genome itself is not even or random. So it is not likely that it will represent all the parts. Some may be very large, some may be more than 10,000 base pairs or 10 kb, some may be 1 kb. That means we might lose some of the genome fragments. So ideally, so the plan here for these people is, why not take a smaller one, smaller recognition one? And what will happen if smaller one is occurring every 256 bases. But then we don't want, the idea is we want to clone larger fragments. Then the genome might become very, very small, isn't it? So we don't want very, very small fragments either. Okay, because we want to, we have phage vectors. Why we don't want it very small is you have to increase the clone number of clone size. What I mean by this is, in the previous one, let me go this. What is the minimum number of clones that should be there for this genome to be represented in a gene library? Approximately. We have assumed 6 genetic elements. The minimum that is required is 6. Okay? Assume that each of them is, say, 2 kb. I'm just making up stuff so you can follow. Then the minimum required is 6 clones. That's the least possible. Because there are 6 genetic elements, you need at least 6 of them. If you ideally, that is what is going to be there theoretically. If you have cut this DNA many more times, right, about 256 or 200 base pairs, then what is the minimum number of clones that are required? That will be huge, right? 60 clones or so, at least. So if you have very small fragments, then the number of the gene library size will increase big. That means the number of clones you should have to be able to represent all the genes will be higher. If you didn't understand the point here, it's all right. We will discuss again some other time. I hope you grasped a little bit. The larger the size, the less number of clones we require. Larger size of the fragment that we put in. If you put in very small, then we will have, the problem is that you will have to have a very big gene library so that all the genetic elements are represented. So what these people had in mind is, if I use a 4-base cutter, it cuts into very small fragments. So what they did was use partial digestion of South3a. Partial means you'll only allow for a little bit of time for the interaction between DNA and the restriction endonuclease South3a. So that means they will take a tube, add the DNA, add very little amount of South3a. They wait for some time, and then they will stop the reaction by adding chemicals or cleaning it up. So by managing, one will have to look at how things happen. The time and concentrations of maybe DNA and the enzyme should be optimized. For each organism, every time they're doing, they would have added these at different points, they would have taken and then allowed to electrophoresis. They will perform electrophoresis and find out which fragments, which is the best concentration and best time. They will determine this by experimentation, and then they will be able to figure out what are the conditions required for, time and concentration to have the best partial digestion so that their objectives are met. Okay, so that is meant by that. Why that is the reason for partial digestion and why South3a is used, so that the probability of occurrence is very high. And then maybe in one genome it would cut here, here, and here. You would have another genome in that it may cut somewhere else in a different location. It may cut here, here, here. You are generating random fragments that are likely to be representing the whole of the genome. Okay? And second step is to treat with phosphatase, the alkaline phosphatase. We know what alkaline phosphatase does, isn't it? Alkaline phosphatase removes the 5' phosphate group. Sorry, P5'. It removes the phosphate groups and leaves OH. When we were discussing about small cloning strategies and so on, it is always we used the alkaline phosphatase to treat the vector. Why were we bothered about it? Because, say, I'll just try to take it up here. We have a plasmid with BAMH1. We have fragment with BAMH1 overhangs. We digest it and then ligate. What are all the possibilities? The ideal possibility is that we get the gene of interest ligated into the vector. But what may also happen is we may have vector-vector religation. And when we transform and see, you will get both colonies, one representing the recombinant, one representing the non-recombinant. This is vector-vector religation. Okay, so we don't want these to be in our plate at all, because the more number of vectors are engaged or involved in vector-vector religation, the probability of the recombinant formation is also getting lesser. Why is that? More and more vector is consumed in formation of this, and the probability of formation of recombinant is also reduced. So what we do is, before ligation, once the vector is cleaved by BAMH1, it would have double-stranded, and we would have treated with alkaline phosphatase. I'll just write as alkaline phosphatase so that the phosphate groups are removed. So if you look at the DNA like this, and I'll put this as the 5', here would have been the phosphate group. This is phosphate, OH, and OH. The ligase would have made this, would have continued, would have made the ligation reaction or formation of the phosphodiester bond. And for formation of phosphodiester bond, we want a 5' phosphate group and 3' OH. Because we are treating with alkaline phosphatase would have removed the phosphate group, right? And the same thing would have happened here too. And when it removes, it will leave a OH. It will leave OH here. And now ligase cannot act upon it. So what we are doing by adding alkaline phosphatase is we are reducing the formation of vector-vector religation. So there will not be any recombinants, any non-recombinant colonies from there. When we do this, we are also increasing the probability of formation of vector fragment ligation. So in majority of the cloning protocols, if you see, it is the vector that is treated with phosphatase. Contrary to that, in this protocol, we have it different. What we are doing is we are treating the fragment that needs to be cloned with alkaline phosphatase. That is something odd. Okay, and I'll try to describe why. So if you remember the objective of what we are trying to do in this, it is gene library. It's not simple cloning. What is our objective in gene library? We want to say, in the previous one, I have given you that here is A, B, C, D, and so on. But that's not how we work. We are going from answer to the question. Usually what would have happened? We don't know I gave you a sample, and the sample is genomic DNA. I want you to find out all the genes, the distances, the order, right? Why do we construct genomic DNA? To know about to characterize the genomic DNA. What do you mean characterize? we should be able to find out what are all the genes and genetic elements present in it. First point. We should be able to, second would be the order of the genes. Which gene is coming after which gene, right? The sequence of genes, that is what I meant. Sequence of genes, I said order so that I want to know which gene is after A. Is it B or C? I mean, is it B, C, or which gene is present after B? So one should be able to determine A, B, C, D, that the genes are A is present upstream of B, B is present upstream of C, and C is present upstream of D. That order of genes is what they are the goal of genomic library. The third is to find out the distances. How much distance is there? Right? Eventually, we would want to sequence the whole genome itself. Only then we are going to find, right? Sequence the whole genome. So gene library is, there are multiple things that we need to remember. So we need to know all genes. That's why when I'm defining gene library, I said each of the genes should be represented at least once. Then we should know the order of genes, the distances between them, and eventually, of course, the sequence. When we sequence, we will know all the three. How can we do that using gene library? If you have cloned A in one, right? A is here and B is here. How do you get things? The idea is to make overlapping fragments. So somewhere we will clone something like this from here to here. In another fragment, maybe we have cloned like this. In another one, maybe like this, and so on. So one of it may have, say, A and D. We will do some problems later time, and we will get to know many things then. Something like this. If I constructed A, B, B, C, C, D, if I knew those, I can align them and determine the sequence. A is there. A must have been upstream of B based on what we see here, and which must have been under C and D. That is how we can find out the sequence, the number of genes, the distances, and the order as well. This is the basic concept. There will be many other things. I want to know if you have learned about chromosome mapping, right? Did you do it using conjugation? You can do it using conjugation, transduction. These are classic examples. If you are interested, you may read Genetics by Payers. There are some of the problems well explained and illustrated. You can try it. And these questions usually come for CSAR. Not that these are very important technically at this time, but it is a skill of deduction. It's a knowledge or intellectual skill. And that is why they still ask in CSAR and so on. And that's why we still read them as well. If you haven't read it as a part of microbiology or genetic engineering or molecular biology or genetic engineering, we can take this up at least one class, so that I'm hoping that it will be useful for you in GATE and CSAR. Okay. So in gene library, this is the general concept. They would have cloned each of them, then fragments, they would have made random fragments. They would have taken each of them, sequenced them, and once they sequence it, the contigs or fragments the sequences could be lined up like this, and then they would have determined, and eventually they would have found this as the answer. Okay. So I have gone to this explanation to explain just one concept. Why are they treating this fragment, the genome fragments with South3a? What is the next step at this stage? It is ligation and mixing, right? Once the vector is cut and fragments are isolated, we are going to use ligation. There is a possibility that in the previous one, there is a possibility if I'm doing if I add a ligase, I'll put these so that it will become easy. I'm trying to use it differently. Don't worry. I, J, K, L. These are the genes, and assume this is the cut fragments. Okay, I wouldn't want to call it as genes. And if I add ligase at some point, right? I'm adding it here in this step. This is ligase. What may happen? As always, you should always think of all the possibilities. There is a chance that A and B can ligate. There is a chance that A and C can ligate because their overhangs are identical. And D can ligate with J. And then it could get cloned into one of the phages. But actually, are they nearby? A and B are nearby. Okay, we are going to the answer. Okay. If we think that A and C, we cloned it, we sequenced it, and we can say that A and C are together. But that's actually a mistake, because it is that these fragments have gotten ligated as well, right? Somehow we should have a way to prevent these fragments from ligating with each other. No ligation. We don't want any kind of ligation among the fragments of the genome, because it will complicate the study. It will actually mislead us from what is the actual sequence of genes on the genome. That is the reason why we are treating the fragments with phosphatase in this particular case. In general, you will find that the vector is treated with ligase. But in this case, we are treating the genome because of the objectives for genomic library. Did you understand, or you have any questions on this? Okay, if you didn't have, we'll move on, and you can always have a look at it and get back to me with any questions. Okay?
Yes, sir.
Yes, good. So from then on, that is the explanation for many things that are happening. That is the new part that we are discussing today with this protocol. There is one more small point here, is size fractionation. That means once you have digested with partial digestion, we have discussed about why EMBL3a is used. We discussed about why did we do partial digestion. We also discussed about why are we treating it with phosphatase. And the next part is size fractionation, which is something like pulse field gel electrophoresis. Because these are size fractionation would simply mean you're performing electrophoresis and trying to, based on their molecular size, they would be traveling and they would form different bands. And you take the band of your interest, purify, and do whatever you want to do. That's meaning of size fractionation. And here we want as large as possible fragment, right? But there is a limitation. About 20 kb can be cloned into this in here. 20 kb is kind of limit. So the fragment sizes that we want should be around 20 kb. 20 kb is a very large fragment. So we cannot do it in traditional or the simple electrophoresis. I hope you remember that size of the fragment or the size of DNA, if you electrophoretic mobility, right? It's the movement of DNA in an electric field. Size of DNA, mobility, if you say, mobility of the DNA, one second, mobility of DNA is based on size. We don't count charge because it's uniform, and size is one thing. I mean, because the DNA is uniformly charged. Larger fragments will have larger negative charges, smaller ones will have smaller. It is basically dependent on the size anyway. The second thing would be concentration of agarose and the electric field applied and so on. So concentration of agarose is inversely proportional, right? That means if you have higher concentration, then the mobility will be very slow. And we are talking about sizes that are 20 kb. Normally, we are doing about fragments that are less than 1 kb, maybe 5, 10 kbs. 20 kb is too huge. In that case, we cannot use normal one, because if we want to, because the size is very high, we'll have to reduce the concentration of agarose. If we reduce beyond certain time, then you cannot, it will not form a proper gel, right? We need the gel to be stable enough to hold the DNA. And the other point is that during electrophoresis, there is also increase in the temperature. When there is increase in the temperature, the gel, if the concentration is, agarose concentration is very less, it might actually dissolve also. So it is a complication. For that, we use the pulse field gel electrophoresis. I hope you remember it. You will have, there are different configurations of pulse field gel electrophoresis where you can have something like this. Some of them could be reverse also. Some of them may have even more number of electrodes as well. So the DNA gel is somewhere around here, and samples are loaded in the wells. And at one point, you may have this set 1 could be active electrodes, plus and minus, or positive and negative. So the DNA will move something like this. And computer will switch it off and turn on, say, sorry, this could have been the 1. This is 2, 1, and this one is 3. So at any given time, 2 different electrode sets could be active. So either 3 and 3, so the direction of travel, sorry, it should be in the reverse actually. This DNA is negative and it is traveling towards positive, okay? So I will correct it before we make any further mistakes. Let's check. I had an animation in one of the PPTs, maybe I will share it with you, and that's a good animation to represent that. So pulse field gel electrophoresis could be in different ways. I'll just try again, in which you may have electrodes 1, 2, and 3. 1, 2, and 3. This will be the, on the top, we will have the negative charge, minus, minus, and bottom will have plus charge. And this is where we put the agarose gel, and we load the samples here. And at any given time, we'll have, say, we'll call it as 1. Electrode sets 1 will be active, so the DNA will move like this. Then it could be electrode sets 2 will be active, then the DNA will travel this way. And at a later time, electrodes could be in 3. So the DNA will be moving like this. So basically, the concept of pulse field gel electrophoresis, sometimes there are also reverse ways of exactly reverse also. That means at one point you may have minus and plus here, sometimes plus and minus here for the second thing. So the DNA can move a little bit up, move a little bit up, like that, reverse and move forward too. There are several configurations of it. Different people have tried and used. So I'm not going into those details, but the point is, during pulse field gel electrophoresis, you can get even large fragments such as 20 kb fragments as well move or separate based on mobility without compromising too much on the concentration of agarose, because that is essential to keep the DNA. Why are we using the gel in the first place? Why not just have it in the liquid? Then we are not going to get the DNA in our place. The DNA would have just traveled here. Our objective is fractionate the DNA based on sizes and isolate them for our further purposes. Right? So that is the importance of pulseful gel electrophoresis. A simple phrase, size fractionation, means that. But if you are typically in an electrophoresis, in gene libraries, they perform that. In this case, they said it's not required. Why is it not required? Anybody? The reason for it is the head particles. When we perform in vitro packaging, this is a step that we will discuss some other time, where for in vitro packaging, we said that we need cost site. That is essential on both sides. There should be cost site for packaging. Second thing that is important is size should be between, say, 38 to 53 kb. Anything lesser than that or larger than that would not fit into the head particles. So if we said that left arm is 20 kb and right arm is about 9 kb, how much more can be put in? Maybe we can put around 22 or 23. Is that right? Something like that, approximately. If there is smaller one, so all the fragments that are within so this is 29, the vector fragments alone. So how much did we have? So about 24, right? Maximum of 24 can be put into it, and a minimum of 9. Yeah, a minimum of, say, 10. Somewhere between 10 to 24 kb of fragment can fit into in between and then still be packaged. That's the important thing. Okay. That is why they say size fractionation is not required here, because we already incorporating it in the as a part of the space constraints of the head particle during in vitro packaging. But otherwise, pulse field gel electrophoresis is used in many cases to fractionate large fragments. It's better and easy also, so that we don't have complications. Anyway, I hope you understood the many things that we have discussed. Why the protocol? When you are looking at a protocol, don't mug up the protocol. What makes you better as a scientist or a genetic engineer is that you know why something is used. It's not necessary that we all remember every detail, but at least when we are learning something, or make it our strategy that we try to know why that step is. Everywhere, why is the important thing. You should be able to ask the question, try to find out the answer for it if we didn't know. Not that I don't remember every detail about every protocol, but at least when I was doing, I try to know at least why that is there. Because we are learning these as a course right now, I would want that you try to remember, just don't mug up that it is South3A is used, phosphatase is added, and so on. It doesn't make sense that way. Because if you really are going to implement anything like this, say you are the person doing this protocol, following this protocol, you didn't have something, like say for example, you didn't have South3A or phosphatase, then what should you do? And if you didn't use South3A, if you didn't use phosphatase, what should you bear in mind when you get the result, right? So these are the things you will troubleshooting. You are solving problems. That is an important skill about being a scientist. What all scientists do is you have uncertainty, you don't know what it is, and you're supposed to know it, right? We discussed this concept sometime. You don't know what to do. That is means it's a life of troubleshooting. Basically, scientist is life of troubleshooting. You don't know, you want something, you have to get to there. How would you approach it? And in that sense, you need to know each of these steps. Okay, so I'll just quickly rewind what we have discussed in the previous class and now. So we have, now I'll just focus on the vector part. This is the left arm, right arm. Then we had these polylinkers, red and GAM for spy phenotype. We digest it with BAMH1. We are choosing BAMH1 because we can use South3A. If we use something else, we should have found some other restriction endonucleases. There are many sets of endonucleases, but BAMH1 and South3A are common. The vector we are treating with BAMH1 and EcoR1, these two, that is for a purpose, because we would want to remove this polylinker once upon cutting, so that the stuffer region will not be able to ligate with either left arm or the right arm. We want to limit, or we want to get rid of, we don't want it to ligate. And how are we getting rid of these small fragments? By using precipitation, we can get rid of it, because the larger ones will form pellet fast and the smaller ones will be here. And when we remove the supernatant, the polylinkers will fall off as well. Then we have three parts: left arm, stuffer region, and the right arm. And then we have made these South3A fragments, and we have treated with phosphatase from the genomic DNA. We are mixing and ligating. So there are several possibilities. Out of them, most of it will be left arm, right arm, and you may have the several fragments attached, and you may have concatamers of those as well. So, and when we do in vitro packaging is they produce, normally lambda phage packaging would have happened within the bacterial cell. Once the genome is produced and the head particles are there, these are packaged into the head particles. But what may also happen in artificially, what we do is we have made the concatamers here. They synthesize the head particles and they add, there is an order of things to add so that they will form certain phages. So they use helper phage, which has the genes overly expressed for head particles and so on. And then they make the head. They use this DNA and it is usually packaged. Then they add the neck and tail regions and so on, so that virulent phages are formed. And then it is plated on P2 lysogen. And there is also SUP+. We will discuss that at some other point. Suppressor, it can be used as a selectable marker. So we are plating on P2 lysogen because we want to exploit on the part of spy phenotype, sensitive to P2 inhibition. The concept of it is that wild type lambda phage can infect E. coli, the wild type E. coli or normal E. coli, but it cannot infect P2 lysogen. What do you mean by P2 lysogen? It is also E. coli. Only thing is that P2 prophage is present on its genome. So somehow the P2 prophage inhibits lambda from infecting the P2 lysogen. There are two genes that are found out to be responsible. They are rD and GAM. If we have deleted rD and GAM, they can form plaques on E. coli. They can also form plaques on P2 lysogen. Okay, that is the reason why rD and GAM are present in the stuffer region. Because if by mistake or some other ways, if somehow there is a recombinant, if something gets packaged with left arm stuffer region and right arm, because the stuffer region has rD and GAM, it will not form a plaque. Even if it gets packaged into the head particles, it will fail to form plaques. That means only those with left fragment and the right, these recombinants only will get packaged. I mean, they will get packaged, and only those can form the plaques on P2 lysogen. Okay. Any questions on this? Anybody?
No, sir.
Okay, that's all right. Prepare for your exams. When is your tomorrow? When is your exam tomorrow? Do you have an exam tomorrow? I'm asking any exam that you might have tomorrow, any subject.
No, I don't think so, sir.
Okay.
Today we have one.
Yes, then.
I think it's the other electives.
Okay.
I think it's the electives, not any core subjects or all that. So that is only like next week we have to.
Okay. That's fine then.
Okay,
We'll
sir.
discuss